name is Anton Kulaga. I am from computational biology of aging group. And as you might guess, today I will be talking about comparative transcriptomics of longevity. So I will be talking about long-living species like whales and what we can learn from the long-living species, how we can apply what we can learn, and how it can be used in further analysis. So you see that here I just have only some set of species, starting from the longest living 211 years bowhead whale and up to very short living mouse. And this choice of species, it's not by accident. And there is one particular whale here who is guilty in this choice of species. It's a gray whale in the middle. And also there are some particular collaborators who provided our group with samples from newly sequenced gray whale. So it is a sample of kidney of gray whale mother and her kid with a sample of liver. And I received it from Alexey Moskalev, who is here. And it was sequenced, and together with our collaborators in Ben Gurion University, we started analyzing its transcriptomic data. And we decided that as we want to know what we can learn from longevity of the whale, it makes sense not to take just gray whale in vacuum, but to compare it with the species that you see from the very beginning. So we decided to compare it with the longest living bowhead whale, also with the whale that lives shorter, like minke whale. So we know what is the signal of longevity here. And we also took, of course, human, because we are interested in human applications. And we also took long-living animal as necked mod red, because we wanted to also separate factors that are common for all long-living animals, not just whales. And of course, for the control, we had cow. So we are not really interested in how whale is similar to cow. We are interested in other aspects. And we took a short-living mouse so we can see what long-living species have in common and how they are different. And when you do de novo transcriptomics, de novo genomics, you have multiple challenges. And if you even look at the genomes, you see that they are highly heterogeneous and that you have a lot of challenges how you can compare them, because some of them are done with some technologies, some are good, some are bad. The way how samples are treated are different. And in our case, we decided to concentrate more not on genomics, but on transcriptomics. There we had better quality data, and there we could see some interesting processes that are happening. So coming to our bioinformatic pipeline, so what we did, we took, for gray whale, we got data from Alexey Moskalev, so it was original data. For all other species, we downloaded public available data sets. And then we, for non-model organism, we also needed some to assemble the transcriptome that we can use as reference. So here you see that we do both RNA sequencing, and we also did de novo assembly of a transcriptome. And then we did quantification. And the challenge that we have is that if you look at something even at a gene level, if here is just an example not of the whale, but of mice. And here just a heat map. So red is like high correlation and blue is very little correlation. And you see that even for for the organism with very good genome and transcriptome, you have high variation when you analyze at gene or transcript level. So between two tissues, it can be like 50% correlation, and it's actually quite normal for many mouse samples. And as we have way more heterogeneous data, we wanted to be more robust because the noise is much higher when you have different species. So what we did, we came from just transcripts to first we predicted potential proteins from coding genes for those organisms like gray whale where we didn't know it for sure. So, so we applied some bioinformatic tools, PFAM, and then UniProt, UniRef clusters, and we aligned it to known protein clusters. So in the end, we got proteins that are predicted to be produced from those transcripts. In this way, we went from DNA, where we can have synonymous mutations, where that is way more variable than proteins, because there can be introns, then can be synonymous mutations, and so on, to basically comparing expressions by the proteins to which transcripts are aligned. And then we started looking into different gene ontology processes and different pathways. And there, after aggregation by, so it's actually example not with whale, but with same actually data with mice. And you see that when we went to gene processes, you see that correlations like 98% for same tissues and 81% for different tissues. So of course we lose some sensitivity, but biological processes are way more conserved, and if we have a lot of noise in the data, it's better to be less sensitive but to be more robust. So if we look into our, so it's a correlation matrix, like basically same species on as rows and columns, and the diagonal is 1. That means that it's the correlation is 1, and between, well, gray whale with gray whale, of course, it has 1 if it is same sample. And you see that the whales here on the top, they correlate a lot because they are very similar, and they are more distant from human and from other animals. So human is somewhere here, and of course it's so cow is on the right, and for cow and for mice we actually took 2 different samples. And here you see that here the correlations are actually closer to relatedness to animals. So cow is closer to whales, whales are closer to each other, and so on. But when we switch from just looking at all gene ontology processes, all common pathways, to categories that are important to aging, and here we actually see quite different picture. It's probably too small to see from here, but here I sorted the animals by their lifespan. So we have on the left, we have bowhead whale, left column, then the second column is human. After human, we have minke whale, we have gray whale. After gray whale, we have minke whale. And on the right, of course, on the very, very right, we have mouse. And you see that, that here we have high correlation in between species that are actually distant. Human is very distant from whales, but you see that it's second column, and it shows similar patterns for longevity-related biological processes. And then we started to look into categories by types. So for DNA, this is a picture for DNA repair, I think, and it's in liver. And again, you see that the left part, the long-living animals, including human and necked mold rat, it is way more red than the right part. And in right part, we have cows that is very similar to whales, but you see that cow is blue. It's very, very far on those processes from whales. It means that there is a big evolutionary selection for longevity in those DNA repair-related processes. And then we looked into autophagia, and here you also see, so the sorting up is again with the age, and there you see that left, including humans and neck mold rat, it is more red, and cows, cow is almost blue everywhere, so it's way further than whales. And we also see same patterns. Well, for hypoxia, it's not that straightforward. So for hypoxia, we have some, well, and for some processes, we see that, for instance, for negative regulation of hypoxia, humans is blue. It's doesn't go together with whales and neck mold rat, but we still see the pattern here. And probably the most interesting here what we have, and I'm thankful to one of the previous speakers that told about ubiquitin and protein turnover. That basically you very clearly see a pattern that with some small exceptions, everything on the left is red, including human and including neck mold rat. And that means that for long-living animals, it's important to maintain protein turnover efficiently. And we also analyzed pathways. Here the ordering is a bit different. So we had human a bit further, but trust me, it in many cases it also goes together. And in cases where there are blue, well, the color is not that sensitive, but in many cases it also goes together. So after looking into pathways and gene ontologies processes related to aging, we looked into longevity-associated genes. So we took those genes from the database, well, basically from GeneAge, the database of João Pedro, Roberto Couto, and many other people. And we tried to look, here we did a little bit other alignments, and we tried to look how those longevity-associated genes expressed in cow and gray whale, and how many of them are actually found. And there I mean, the results are not that straightforward, but we see that for gray whale, we managed to find more longevity-associated genes, and they are expressed way more. And if we, yeah, so that's what we found for legs. And if we take a greater picture of why we are comparing those different species, is that our main aim and our lab interest is finding some interesting ideas and genes that we can, so basically here we have as part of a large map of our genomics project. So here we do homology, different types of homology and comparative analysis. And if we do some analysis in the middle, we can come up with some ideas that we can test on model organisms. And yesterday, for instance, Alexei suggested to try something on whale cells. And that means that we can prototype some interventions inspired by whales on model organisms. And with this, I come to conclusions. So here we tried to look into very different species. And it happened that despite a huge amount of noise that we have from highly heterogeneous, highly different data, some of which of different quality done with different instruments, we still see that we see common patterns that are longevity related. We see evolutionary selection of pro-longevity gene processes, and that is often more important than relativeness of animals like whales and cow. And we also see some patterns with longevity-associated genes. And now we are continuing our work to actually try also other bioinformatic methods to cross-validate our results, and including autoencoders that are used in machine learning. We are also adding more species because we are interested to see how it will work for more species, like 10 or 15 or 20 together, if we can say find something interesting. And currently we are also thinking if we can check something experimentally on what we have found. And on this, thank you very much for your attention, and I wish you all to live at least as long as a bowhead whale.
How many species you using to your work?
Well, basically those that I mentioned. So it is bowhead whale, it is gray whale, it is minke whale, it is cow, it is human, it is mouse, and it is noctimal rat. And for mice, I think noctimal rats and cows, we tried to get samples from different experiments to control for a batch effect. That means that what we get, it shouldn't be an artifact that some lab did something in a strange way. So we see that the patterns hold even from very different experiments. We also did some validation of the methods on model organisms. We took a lot of mice samples and human samples, but we had some restrictions for non-model organism. I mean that for gray whale, it was only lab of Alexei who did the work, and we couldn't get anything from another lab. And also we couldn't control for get old whale, young whale, and the same gorgon. I mean, we had young liver and old kidney basically there. And same for other whales. I mean, for non-model organism, you select what is in the literature, or what is on NCBI, ENA, or other databases.
Short question. What about the validation? You may try using not all the species, but for example, 4 species for training and the several species for validate your approach to, for example, predict the maximum of longevity.
Well, I mean, we used many animals, and we selected animals in a way that we could control for relatedness, like whales and cow, that because cows are short-living, they should not have similar patterns to whales on longevity if it is longevity-selected genes. Well, we want to increase more, we want to add more species, it's our aim, and we also want to try to do course validations with different ways how we can align transcriptomes, and with different ways of analyzing the data. So it's next step probably.